we're going to actually talk about the topic now. And before I do so, let's say hello to our guests. So we have with us James Henriksen, who's an environmental microbiologist and bioinformation. Okay, sorry. This is a word I have not.
Bioinformaticist
Bioinformaticist.
or
Man, I knew when I read it the first
bioinformatician.
time, I was like,
Yeah,
there's no way I'm going to say this right.
sorry.
No, that's okay. I want to know what it is. So do you want to actually give yourself
I
a better
yeah,
introduction?
certainly. I'm James Hendrickson, and uh, I was trained as a classic environmental microbiologist, looking at the microbes that exist out in nature, not anything that causes disease. And I'm also a huge computer nerd, and so I teach myself to program. I learned that I am very lucky. I'm living in a time when microbiology has enormous amounts of data from being able to read DNA from organisms in the environment. And so manipulating that would be a bioinformatician or bioinformaticist, depending on which school of thought you come from.
Those are great words. I will put that in my personal dictionary of wanting to know what they are. You didn't make that up though? That was an existing term.
Oh yeah, well, when I first went through grad school, you couldn't really get a degree in this, so I was mostly self-taught. It's now something you can study in school and get credentialed online in. If you are a programmer and you're interested in biology, it's a great way to be able to contribute. Uh, if you're interested in biology and want to learn programming, it's a fun crossover science.
That's awesome. I did not know that existed, and I'm sure some folks will really benefit from that being included now forever in as an actual school of thought. So, and you're also the founder of Two Frontiers Project. Is that connected to the university or totally separate?
It's separate. It's a now international nonprofit research institute, and I'll tell you a little bit about it. I have some pretty pictures and slides, and it's been a wild ride over the last few years, founding a nonprofit group, and then it becoming part of my own work here at Colorado State University.
Oh, awesome. I'm excited to hear more. We also have Sarah Newman on board and also Caroline Nickerson, who I'll talk about briefly too, so she can say hi. But they're related now because they work together on sitsci.org. So, Sarah, as Director of Operations at Sitsci, do you want to tell us a little bit more about what your involvement is for this project and everything else you want to mention?
Yeah, so my involvement in this project has been really fun. I get to wear a project management type hat and help with the engagement and participation part. James and his team have the science dialed in. They also knew kind of what questions they really wanted to ask and how people could be involved in it from the public population. So my job was to come in and help them connect with those folks through sitsci.org, creating a project and a space for them to showcase and spin up quickly, really. This project has only been around for 7 months and we already have 100 participants, which is incredible. So it's been very exciting. And I've worn lots of hats in the citizen science and participatory science space in the past, which you can see on the slide. But yes, we're delighted to have Caroline working with us, and we're always excited to partner with SciStarter.
I'll actually just drop the screen share so we can say hi. Let me.
Yeah,
There
hey
we go. Now we're all
everybody.
together.
I've been looking forward to this webinar. I have been trying to find slime in my house and haven't yet. So I'm hoping to
Ooh.
get tips via this particular event.
It's either everything is super new
Wait,
or it's or we just have not found the location for it. I guess we'll find out. All right, I'm going to go ahead and pass it back over to James if you'd like to share your screen so we can get started in talking about the actual project. But if you have questions during this, as I mentioned before, feel free to just drop questions or comments in the chat about this. I know I will be trying to prevent myself from interrupting, just be like, what?
no, please interrupt. I would love to just make this a conversation. So, Two
Oh no.
Frontiers Project is. Can you hear me?
Yeah, I can now. I'm sorry. I thought you were frozen
Okay.
for a second, but we're good. We're good.
Great. So Two Frontiers Project is a nonprofit research institute that's set up to study to explore and to apply microbial life from the depths of the ocean to the reaches of space. And this entire group, we're only about, it's only been 10 months since we got our nonprofit status. We've only been around even as an idea for a couple of years. And it all came about because an old friend of mine Braden Turney, had this crazy idea that we should go to an island in the Aegean Sea off of Sicily and work with some really great scientists that he knew there to sample these underwater high CO2 vents. And we, as academic scientists often do, we put together a proposal, tried to get some funding because we thought this would be a great place to look for organisms that could capture CO2, eat CO2, because it's one of the very few CO2 seeps on the planet that has light penetrating to it. And we were turned down, but Braden managed to raise some funds to do it anyway, not from the federal government. And we went to this beautiful place, sampled, and with some collaborators who were just working in their free time, were able to isolate this cyanobacteria that you mentioned, that gobbles CO2 very quickly under certain conditions. And it also, which is almost more important, it sinks. And so if you want an organism that could grow in a high CO2 environment, maybe something like the output from a fossil fuel plant, and capture that CO2 using just light energy and then sink so that you could recover that biomass, this would be a great organism that you might want to explore the uses of. And so that experience and led us to think that maybe we were onto something. It actually, we discovered something that was what we had set out to find, which isn't always the case in environmental microbiology, and we decided we wanted to do a lot more of it. And that's when we hatched this idea to start a nonprofit research institute that now has collaborators all over the world. And we continued this idea here in the Rocky Mountains of Colorado. Turns out Colorado has more high CO2 springs than any other state. What you're seeing there in that second image is this mound. It's like 20 feet tall with minerals that have been formed from a high CO2 spring gushing out of the side of a mountain. And all of those colors are alive. They're microbes that are growing in this acidic, carbonated, high mineral, high iron environment. We participated with some of our collaborators from Sicily, Marco Malazzo, among others, to sample some deep ocean CO2 vents. We just got back from Shikajima, Japan, where there's both terrestrial and underwater CO2 vents, and have collaborations in Papua New Guinea. And the idea is to take information, DNA sequence, organisms from all of these places around the world, and create what we're calling a living database, an open source of information for interested citizen scientists, maybe bioinformaticists, for researchers, to examine how life has adapted, what life is there, and how life has adapted to these high CO2 environments. And alongside that work that we've been doing since we went to Volcano on these high CO2 vents, we've also started looking at corals in some of these unusual environments. The microbes that grow in and on corals are very important for their health. In some of these rare, unusual environments, the water is acidic, much like what release of fossil fuels is doing to the global ocean. And yet there are some kinds of corals that survive. And so we're exploring a little bit about how that occurs and what we can learn there to help other corals. We've visited the Mojave Desert to find microbes that grow on plants in this unusual lava field there, to see if those same microbes could help plants grow in lunar regolith and moon dust. And so the general theme here is we go to someplace very unusual, exotic even, and we explore the microbes that are there using DNA sequencing and culturing, growing these organisms. And I'm super excited to talk to you all today about what I think could be some of the most exotic and unusual places that we're going to explore, and that is our collaboration with the Citizen Science Project. Mostly I'll be talking about the extremophiles in your home. So what do I mean? How is a underwater CO2 vent? You could kind of see how that is extreme, places next to volcanoes, but in our home? Well, if you think about it, what we have created in our living spaces is something that is super unusual. And to the microbes that are living there, it's extreme. And and I have to tell you, everywhere you have looked, you can see there's microbes. And I'm sorry for those of you who are germaphobes about
Really
every
hurt me.
surface you're looking at. Yeah. But here's the good thing. They're all harmless. The organisms, almost all of the organisms that grow out in nature, they're adapted for that natural system. You know,
Right.
the very rare microbe that can infect us or cause any kind of sickness or disease, they're adapted to particular temperature, very low iron environment in humans, and other things that are very different from what is in your home. And so while non-microbiologists might be grossed out, it's only natural with some of this stuff. It's everything I'm going to talk about, everything we're interested in, isn't going to make you sick. But they are extreme environments. So if you think about water from condensation on a air conditioning system, it's almost pure water. There's no nutrients there because it's been condensed out of the air. And yet, as it flows out of our house, out of these tubes, and is exposed to sunlight, there can be algae that are growing there. And the very fact that that water is so clean, and yet there's something green there, tells me as an environmental microbiologist a few things. One, it's some kind of algae. There's no plants that can grow under those conditions, and actually even more, I know that it's a bacterial blue-green algae because there's no nitrogen in that water. And the only organisms on the planet that can use light energy and can take the nitrogen in our atmosphere and use it to make the nitrogen that you need to build a cell are cyanobacteria. The next slide or next image down is that slimy kind of orangish pink color. And that's a microbe that's forming polymers in these very low nutrient water.
Can you
Another kind
help
of,
me understand? I'm sorry to interrupt. I just want to know, for anyone who does not know me, polymer, can you describe what does
oh,
that
yeah,
mean? Is
please.
it a one
Yeah,
organism?
polymer is just a chemical phrase. It just means a lot of chemical molecules linked together in chains. And
Okay.
so bacterial polymer is a chemical that's made by hooking things together in these very long chains. So polymers that you encounter every day are things like plastics.
Oh, okay.
And this one is slimier than plastic. It's that is probably a Zooglena organism. Primarily, but it's a whole community. Every one of these images is a complex community of organisms. When I say a particular type of organism, I usually just mean the dominant one or the base of the ecosystem. And so this is a problem because it clogs up pipes for people. But that same capability for an organism to take very low nutrient water and make a polymer has a lot of applications that humans might care about. Um, one of which is making bioplastics, for instance. Some of those polymers have been investigated as good bioplastics. Others are used as thickening agents in food so that your ice cream is creamy. Um, yeah, and actually, I should have mentioned the cyanobacteria, besides fixing nitrogen out of the air, using it to build their cells, they also take CO2 out of the air to build their cells. So they're fixing CO2, which is obviously important for solving human-caused increases in atmospheric CO2. Um, there's a lot of places like this in our house where we make basically pure water and then set up these very unusual environments, like that red color you see underneath that. I think that's
Mhm.
some kind of HVAC device. Um, think that is an iron oxidizing bacteria. So it's an organism that instead of eating sugars like we do and using oxygen, they actually transform iron into rust using oxygen. But of course, there's no carbon to build a cell in iron. So they actually get it out of the air. And this is going to be kind of a general theme. Microbes can do amazing things out there in the environment. And a whole plethora of things that is completely unlike the nature that we can see with our naked eye. So, you're used to thinking about plants that use light energy to fix CO2 out of the air and make carbohydrates, polymers that make up plants. And there's bacteria that do the same thing. there's bacteria that are like us and use all sorts of organic material and combine it with oxygen in order to get energy, and they exhale CO2. And then there's creatures like this that they literally are transforming a metal from one oxidation state to another to make it rust. And then they're fixing CO2 out of the air like a plant, because this is occurring in the dark. And I think that's really cool. And this vast diversity of life, you really aren't exposed to unless you start looking at the microbes. And it's really important for global carbon cycle and nitrogen cycle and other elemental cycles that make up our ecosystem. By looking in the home, it might seem like we're divorced from nature, but for the microbes, it's just another extreme environment, like water coming out of an underwater cave. That makes sense?
I believe so. I'm also thinking, Roland dropped a question in the chat about humidity levels to prevent or promote bacteria growth like this. Do we know what the like levels have to be for this to happen, since we know about like the pure water being such a phenomenon?
Absolutely. So almost all life on this planet requires water, but for microbes, you don't need much. Even in the desert, there can be like microscopic layers of water that allows them to grow on the surface of rocks. So you're never going to get rid of the organism. Now, obviously, we want to prevent our pipes from clogging or our pans from rusting. And there, having enough water to get this high amount of biological activity, you usually do need some amounts of water. So one thing we've noticed as we've started this project is that citizen scientists in places that are more humid, they see a lot more like large amounts of biomass as they look around. Doesn't mean it's not necessarily going on in drier places like here in Colorado. It's just probably a lot less and sort of intermittently as surfaces get watered, or very slowly if there's water is really limiting. So, if you want to prevent this kind of growth in underneath your drip trays, you have to make sure that your drain is operating properly and drying out. And if you work with somebody in the HVAC trades, have a plumber come in to try to solve a problem, that's going to be their solution. And in the meantime, would absolutely love to get whatever strange goo or clogs or rusty, crusty material you can find, because that is life, and it's in these extreme environments.
Oh, that's fascinating. Ooh.
Yeah, a few others that you might not think of, like you think of our hot water heaters. It's like incredibly hot all the time, right? And so you wouldn't think that anything could survive that. But actually, there are microbes there. Again, they aren't the kind that are going to make you sick. They're actually, some previous researchers have found that the microbes that you can find growing in hot water heaters are related to the microbes that grow in the hot geysers of Yellowstone National Park.
Oh, that makes sense.
Yeah,
That's
it's right. It's the same sort of extreme environment. Underground at Yellowstone, your hot water heater in your basement. One is much easier to get to and sample, particularly if you're a citizen scientist.
right.
And but no less important, because that kind of ability to carry out biological processes at high temperatures, that kind of extremophile, might call them a thermophile, it'd be a technical term for it. That is why establishing technology of PCR, establishing technology of genetics was able to be discovered. If you've ever heard of PCR, it allows everything from rapid COVID tests to paternity tests to every kind of genetic manipulation that scientists can do is based on that technology. And it came from an enzyme that's from an organism that was isolated from Yellowstone National Park, one of these thermophiles. So, in your own hot water heater, you might have the next bit of biological technology that revolutionizes genetic engineering.
That is one of the most fascinating things about science, like learning about how things have been discovered is always just such a thrill to see because there are so many circumstances over the years where it's like people weren't scientists way back in the day. They were just like explorers and they wanted to investigate what's happening around them. And half the things we learn just for the heck of learning is foundational for these crazy things or like these discoveries that make big changes or differences like that.
Absolutely.
Yeah.
And I feel like in environmental microbiology, we are still in a stage of exploration. Um, a lot of these samples that folks are sending us from their homes will contain organisms that science knows almost nothing about. And, everybody can actually participate in this kind of exploration, just looking around and trying to find the unusual environments that might have something microbial on it. If if you join the our project on SciStarter, you can look through some of our past forum posts, and Sarah has a couple really nice write-ups about this discovery that I just mentioned, and about that what I'm sure is completely disgusting to most folks, image in the upper right, which is a shower head. There's a Halloween special on that kind of material. And again, it's microbial, doesn't cause disease, but those organisms are making these polymers, these things that are potentially useful. In this, the top of this toilet tank, you can see a little bit of red, little flocks there, and that we think is probably that's a related kind of iron oxidizing microbe, like this rust one that I talked about earlier. And these kind of materials are really important. They're actually nanostructured iron, and people have been exploring some of those materials as a way of cleaning up heavy metal waste from mines, because that red flocculent material can bind heavy metals. So there's applications that help solve human problems in all of these unusual places.
Wow.
And what we're
someone
really, go ahead.
Sorry, Robin just mentioned in the chat that she has that in her home, which is crazy. So that actually leads me to a question that was about all of
Please
these.
join.
Yeah, please join. Please join, Robin.
Please join. We would love to have your observations as a data point, because we are selecting a subset of these observations. If people are willing to sample them for us, we send out kits. And when we get them back in the laboratory, we carry out next generation metagenomic sequencing. So we read the DNA of everything that is there in order to understand it. And that's how we're going to discover if there are these organisms that are unknown to science.
Yeah, I was about to ask, like, do we expect to see similar species? Like the fact of Robin saying that one specifically, or it looks like that one, are we expecting it to be the same species or at least genus or something, family? And then, or are we expecting to find
Great
everything
question.
different because it's so vast?
Few of these home environments have been studied a little bit, usually using older technologies, not being able to sequence the DNA, which is very limiting. And using that more limiting technology, for instance, in hot water heaters, people found some commonalities, like similar genera, similar genus of organisms, but definitely some new species. For some of these other environments, no one knows.
That's so
Like
exciting.
they're so unstudied, I really can't tell you. I'm going to jump. Well, I'll tell you a cool story that I am particularly interested in just a few minutes. So this general idea of working with citizen science so that we can document all these unusual places with a subset of them, because metagenomic DNA sequencing is quite expensive. We're going to work with citizen scientists to collect them. We try to grow organisms under different conditions in the laboratory, and then we read the DNA of everything that's there, both the organisms that we grew and the original sample, and really exploring this with citizen scientists and is the goal of this project. You've met Sarah and I just wanted to talk about some of the other folks who are in the laboratory and on this project. There's Braden and Krista, two good friends of mine who together started Two Frontiers, and then a whole group of really bright undergraduate students who are helping with all of this scientific work in the laboratory when you send us your samples. but really, of course, the stars of any citizen science project are the scientists themselves. And as Sarah alluded to, we've had a really great response. Even though this is a relatively new project, we now have over 100 members. And we even have a handful of observations that are from international, that aren't from the US, that are helping us understand what's out there, which for some of these kinds of microbial growths, there's nothing in the scientific literature at all. So we're really building some foundational knowledge here. Few more examples here, and I just want to jump to the bottom two since I've talked about some of the others a lot and what they do. What you're looking at, those bottom two images are from two different citizen scientists on opposite sides of the country, and they're in their dishwashers. And when people make an observation, we have a whole series of questions that we ask. Sarah will show you a few of these later. Each one gives us a clue as to what kind of microbiology might be going on. They also help us compare between observations. So these two different citizen scientists took photos of their dishwasher, described it. Their descriptions matched almost exactly. When you look at these two photos, they have the same growth in their dishwashers on different sides of the planet. And so this gets back to your question. We think they're related somehow. Is there some place that's like a dishwasher with cycles of hot and cold and detergents somewhere in nature that they've colonized? Maybe they came from the factory and they're really related ecosystem of microbes. No one knows. I have no idea, but I do know this. If both of them, actually one of them has already sent us a sample, if we get the sample back from the other and we look at their DNA, we're going to be able to tell the whole constellation of microbes that make up that community. We're going to be able to look at the dominant organisms, and we're going to be able to see if they're related and how closely they're related. And that will tell us if this is something that is a single community that has been separated only recently, if they're two totally different communities that for some reason happen to have the same macroscopic presentation, or if they're different, so they have to be coming from different places, but they've colonized these two dishwashers on opposite sides of the country. We'll be able to find out. And that's what's so cool about this project is we're learning things that we didn't even expect. We didn't expect dishwashers to be some place that people would find a lot of microbes. And yet we've found a lot of observations of these places. Okay, very quickly, I want to show you some videos of some of the samples that we've gotten back. So what you're looking at here is microscopic imaging of a sample from a humidifier filter. And you see there's a sort of cubic structure sort of in the middle. That's actually a bit of some mineral that's precipitated out on this humidifier filter. But what I want you to look at is all of these tiny little jiggling dots. Every single one of those is a bacterium that's growing on this filter. This is a sample that came from a rain barrel. And again, these small translucent dots and rods are all bacteria, but if you look closely, there's one zooming about. And those are organisms that are motile. They have various mechanisms by which they can swim very rapidly. And if you looked at the sample, it just looked like clear water, looked like pure rainwater, but yet all of this was going on. You can look at it for it.
They look like they're having a party.
Yeah, or a race. So this one is to me a really awful image, and that's because of this large creature, microscopic. You wouldn't be able to see it with your naked eye, that's swimming around. So it's a paramecium, and these colored fibers you're looking at are fibers from the filter. But again, if you look carefully, there's a lot of these small dots that are bacteria. And the reason I say that it's a really terrifying, horrible image is because that paramecium is a predator of bacteria. It's swimming around, gobbling up the poor defenseless bacteria.
Poor bacteria. That thing is moving too. That is
I know, it's just hovering them up. It's like a giant lion if you're a bacteria. You wouldn't want it to pounce on you. So So we've talked a lot about this Extremophile campaign in your home. How can you participate? We'll walk you through some of the individual steps later, but this is an active project, and we would love to get your observations, your things from your home. And I want to tell you that this audience is getting a special first announcement. Has been announced to anyone else, but our second campaign, which is Extremophile Campaign in the Wild, which lets you, if in your local area you have some of these extreme springs and high CO2 springs or hot springs, you can let us know about those as well. And similarly to the In Your Home campaign, a subset of those, we're going to be sending out sampling kits and sending out kits to ask citizen scientists to take some water measurements and send them back for DNA sequencing. So here's how you would go from SciStarter to make these observations, and I'm going to hand it over to Sarah to walk you through a little bit about what this actually looks like in our remaining time. But thank you so much for listening to me talk so much about all of these organisms that I care about a great deal and think are really important. And I know that you can help us with this project.
Thank you so much for indulging my questions too. That was really awesome. I loved
They were great
I love when
questions.
people are super excited about these things too, because it's very clear that you know what you're talking about and you just really love what you do too. So it makes a difference to be able to understand like why we should care so much. So thank you. Yeah.
Very lucky.
So thank you, James. It looks like there's a question, if I can address that one, Emma on Facebook, about where can I send the bacteria sample for testing. So the best option is to take an image of it and then upload it to the project. And then for us, if it seems like one where there's enough biomass and it's something we could do more testing on, we'll contact you and then send you a sampling kit and send you all the information about how to do that then. So that would be the method I would use. And then these steps are here. So from SciStarter, you can use the visit button and then get to the Extremophile campaign in your home. And we've got some instructions on how to get started there. So you can do that to take the photo. So the main steps are find it, image it, upload it, and answer a few questions about it. And then from there, we decide what we can sample and what we can't. Some of the sequencing is kind of expensive, so we have to be a little particular about what we pick, but we've seen so many cool things already, so encourage you to do that. All right, we can go for it, I think, Emma. And maybe one more. So, okay, perfect. So when you get to SciSci, that's where the project is hosted, and we have a variety of resources that I just wanted to point out to, in addition to the amazing information James has given. We've got a resources tab there that will have some materials to give you more background about what we're talking about, and some articles that people have written about it, and then something called a Places to Observe document. And that is essentially a slideshow of cool places in your house that we think you might find something interesting to scrape and document and photograph. And up above that, here on this slide, you'll see some examples of things people have taken pictures of already. And you've seen some of those from James earlier. I think that middle one is a toilet, inside a toilet tank. And they've got some stuff growing in there. And when you click on some of those things, you'd be able to see them a little bit more up close. And then, so I definitely encourage you to take a look at that Places to Observe, because it does have a lot of cool places in it. And then we'll show you some of what we document in there in a second. So next slide, Emma. So, for example, the hot water heater, like James had talked about earlier, this is an example of one of them. And we've got a little bit of information about where we're thinking you might find something interesting. So in this case, if you drained your hot water heater a little bit, which a lot of people do annually as part of home maintenance, and you caught some of that water in a test tube, you might be able to find something interesting in there. So lots of different places. And next slide, Emma, I'll show you a few more. So this is a screenshot, an example of what a data sheet looks like. It's not the whole data sheet, but it's just gives you a general sense. So we ask for a zip code for the location, just so we generally know where you are. And then a couple of key questions about microbes. Go ahead, James.
Oh, we're doing that to get some kind of idea of the geographic spread of these kinds of microbial growth, but we're keeping it to zip code so that, no one has to know that you have the weird slime underneath your air conditioning.
Exactly. We don't want to embarrass anyone, that is for sure. Although we've had some very excited volunteers say, I've got lots of weird things. I'm happy to share.
It's true.
So it's great.
People after my own heart.
Yes, exactly. And so we ask you a few questions about what you're seeing. Like James said, things like color, smell, amount. Is it something that keeps growing back no matter how much you try to clean it? Those are all really key because James has so much knowledge in his head that it helps him kind of pare down what it might be and why it might be interesting. So definitely answer those questions. It's super helpful. And then the question I have kind of blown up here on the right is, what part of your home did you observe? And so here's that kind of wish list of places, and the red arrows right now are pointing to places where we'd love a few more observations, or maybe don't have any at all yet. And so definitely looking to everyone to kind of check those places out first if you're comfortable doing that. Inside things like your condensation tube for your home heater, or that air conditioner drip tray, also like an ice machine drip tray or possible tube or line, if you can get to those. Certainly if you have solar panels, that would be really interesting. And then like rain barrels or gutters, and then other. So there's definitely been some unique places. Somebody sent us an image of something inside the engine of their car that was growing, and I was like, never would have thought to include that, but awesome. So be creative, and as like I like to think of this as a very low risk kind of opportunity. You can always upload photos if you're not sure. It doesn't hurt anyone in the process, and it's super easy. And the best thing that can happen is you discover something amazing. So if you think it might be interesting, go for it and upload it and have some fun with it. And then in the upper right, I have a note reminding me that we will keep this project open ongoing, but we are hoping to have as many samples as possible by April to do some of to get started on some of the sequencing work. And so certainly don't twiddle your thumbs too much on it. Get going, get out there right away, and have some fun exploring. The other thing I want to mention is while this project is designed more with grown-ups in mind, we had a really inspiring middle schooler reach out to us early on and said, I want to do this as part of my science fair project. And we're like, you go, girl, like, let's do it. So please encourage your the young ones in your life to do a little investigating with you and take a look. Always, of course, safety wise, like have the adults do the data entry, and don't send us any photos of your cute kids, even though we'd love to see them. We always want to protect their safety. But if you want to send a picture of yourself sampling things, those are awesome. We love to share that kind of thing with the community. So we encourage you to do that. And I think that might be my last slide. I can't remember.
James is actually in control. James, do you want to hit the button?
James, is it me or you next?
Yeah, let me. I just kind of reiterated the slide that we had earlier because I wanted to talk a little bit about where to sample and what to look for. How can you tell it's microbial? One, if there's high moisture and you see odd color, goo, sticky stuff, crusty things that build up over time, those are all signs of microbial life. In places where you might expect water to be very clear, like in the hot water heater, if you give it a rock and you pull some water out and there's any kind of cloudiness or coloration to it, that's probably these thermophilic organisms growing there. And I guess in general, things that look gross or, are things that actually is an important part of the natural world. There are these microbial communities that are all around us, just usually they're in such small amounts that we can't see them on, recently cleaned surface. There might just be a, few dozen cells per square centimeter, maybe a few hundred. Um, when you see these mats, it's billions and billions of microbial cells um, that have built
Okay.
up.
I love that rule of thumb. If it looks gross, sure.
Then exactly, and of course this, we do have some restrictions. We talk more about this. Like we really don't want anything that has human bodily fluids or
Oh,
any kind of animal carcasses or anything like that, because I study environmental organisms and things that interact with CO2, not things that interact with the human body or with animals. Um,
yeah.
and but outside of that, there's really a lot to be discovered, and as I said, I'm learning about new kinds of things that are in the built environment from our volunteers all the time, and uh, often with this metagenomic DNA sequencing, it takes some time to get the data back. Um, but even now, just from the observations in the microscopy, I know there's some really cool things going on that could have applications for things like CO2 fixing and uh, environmental cleanup that I think we have a lot that we can draw on from the microbial world. Um,
Before we move on, actually, I did want to ask, which let me check if it was on here. I wanted to ask about the solar panels. How are
yeah.
they? Yeah, like why is that a good location to look for these?
Well, it's some of these things are on here because there's just a hint in the scientific literature. So some researchers uh, swabbed solar panels and looked for some organisms there. And yeah, thank you. That was a great question. And they again didn't sort of apply the suite of tools that Two Frontiers uses, both the culturing and growing of organisms, which is very traditional, but we do some really innovative things with it that allow us to grow more organisms than have been possible in the past. And a lot of these older papers just they didn't do the deep metagenomic sequencing. They maybe looked at sort of DNA barcodes of organisms, which tells you sort of at the what genera, what genus of organisms are there, but doesn't really tell you what they can do. So there's this paper about solar panels, and when I read it, I'm like, well, that's interesting, but I want to know more. So it's in a really extreme environment because it's usually dry, or at least cycles between wet and dry. It's high UV, often higher heat than the surrounding roofs, and they're glass, so it's not like there's any kind of crevices or nutrients that can be extracted like from some of the minerals that are in other types of roofing. And this paper, said that there was a lot of different spore forming microorganisms. So spore is a reproductive structure. It's like a seed that's very resistant to temperature and a lot of chemicals and UV light. You could see how it might sort of settle out of the air and just be there as an inert organism. But they also had some hints that there was a lot of organisms that could not form spores. And what are they doing there? If they're pigmented, then over time they'll be blocking light on your solar panel. Particularly in places that don't get a lot of rain to wash them off. So we'd like to know about that if biofouling is something people should be concerned about. But this, for me, the extreme nature of this sort of high UV exposure with very low nutrients is interesting because there are bacteria out there that can uh, repair their DNA even when there's very high UV light. And I think that there's some interesting things to be learned from those organisms. Some of the compounds that they make to protect themselves from that constant light. Uh, People have talked about making coral reef safe sunscreens out of some of those compounds. They're biologically produced, they break down naturally, but yet they block the UV light for these organisms. And so I'd love to get samples from a solar panel, particularly if yours is very dusty and dirty because it's a low biomass environment, which makes some of these methods very challenging. And so someone out there time you're up on a ladder, you notice your solar panels have a lot of material on them, or maybe they've been there a while. Boy, I would love to see if what those organisms are, what they're doing, and if they have some useful characteristics that might help people.
Yeah, it's interesting. Especially since it's low biomass, like sampling of that later on, if it is something that you want to get the sample on. I was thinking to myself, is there any version of this in which you're really excited about the location in which something is found, but it's just so limited in amount that it's not like sampleable at this point? Is there any reason to prepare someone to learn how to use like a petri dish solvent to like grow it over time? Would that make any sense?
I love your idea there. That's you're really you're thinking like a scientist. That's a great, clever solution. And maybe that would be a great idea for our next citizen science project to, send out materials. And I use petri dishes, and some of the media, the chemicals that I put in them, uh specifically grow organisms that have some of these characteristics. Like there's no other source of carbon, and you have organisms growing, they're fixing CO2 out of the atmosphere. All life needs carbon. So it's not just a way of amplifying a particular organism or a particular characteristic. It actually can be a really important data point. And what you've just described is something that actually Two Frontiers is doing as part of our scientific research. When we go to these volcanic islands off the coast of Japan, we bring along kits that are basically entire microbiological laboratories in a box. Some of
Nice.
them are a bit bigger than others. Ugh, that one's full of different media. So we have this system so that we can, on the spot, on these islands, generate the conditions to grow a particular organism. So in that suitcase, I have everything I need to make about 4,000 different kinds of media to grow different kinds of organisms.
I didn't even think about the different needs for the medium to be different depending on the organism. So that's good to know too. It's not something you can just show up at a grocery store and find. I don't think they sell petri dishes at a grocery store. Never mind. That didn't make any sense.
You'd be surprised. When I worked for biotech startups in the early stages, you can get all sorts of things from a grocery store to do microbiology. Agar is sometimes sold there, which is what's in the petri dishes. But you do want to be a little careful, because if you use material that's very nutrient rich, if you grow them close to body temperature, then you could start to amplify organisms that might be pathogens in the environment. And so this is something we think about a lot and always avoid in the laboratory and in the environment. But I think with citizen scientists, there's a DIY bio community of folks who do microbiology in their homes, and they've come up with a lot of really clever things to be able to do that, be able to grow organisms safely. And I love this idea of starting to take some of these things that we do as scientists and when we're doing field work, and start to roll it out for citizen scientists, and really be able to amplify our reach and folks' ability to use that kind of technology.
Man, I could talk about this for multiple hours, germaphobe or not. But unfortunately, we
I
only have
hope
5
I've
more minutes.
converted
So I
you.
want to make sure we get through everything that you wanted to get through. And I think I pulled your slide down just to see if we could see the box, but we switched gears. So I want to make sure that you didn't want to go back. Do you want to go back to the slide?
Nope. The next thing is really just asking questions and letting you talk about some of your SciStarter events that are coming up.
Oh, that's right. Oh, yes, just my slides. Okay, gotcha. Well, thank you so much, both of you, for explaining how this all works and how it's begun, and also the deadline. So, Sarah, just to confirm, by the start of April, you want pictures, observations sent in? Is that right? Or like throughout the month of April is okay as well?
I think throughout the month, James, correct me if I'm wrong, is totally fine.
Yeah.
And truly after as well. It's more that we're excited to start doing the next phase. So we will get to do that more around that time. And so we'll keep it open. We will continue to collect observations ongoing. Who knows when, like we've had some amazing support funding wise for this project. And so the more excitement that's generated, you never know when another kind of opportunity will come to maybe do a little more sequencing.
Yeah.
Yeah.
So definitely don't let the time stop you. But if you want to be in that first cohort, April's a good time to aim for.
Awesome.
Yes.
That's very good to know. And if someone has questions on the website for sitsci.org, I believe there's a spot where you can ask questions. Is that
Yes.
right?
Yep,
Okay.
definitely.
Yeah, because I apologize. I asked all my selfish questions, but thank you to those of you who did send in your questions, like from Facebook. Thank you, friend, who sent that in. Charmaine, thank you, and others. And if you do have more questions, as I mentioned, you can always reach out to us too, and we can forward it on to these folks. And on the sitsci.org website, you can find it as well. I'll go ahead and share my screen again, just so you can see some very simple ending slides, just to reiterate. We don't have our official schedule out for April or March and April events, so just be on the lookout. You can always look at our blog for updates. That is SciStarter Live, and our event database. Of course, we'll have those events as well. So just a heads up that if you are like, this is awesome, yes it is, look for the next one that'll be eventually on the list. Unfortunately not ready for now. I asked about April too, Sarah, because I really was hoping we could make sure this was a part of Citizen Science Month. So yes, it is. If any of you participate in this project during April, it counts as part of our 1 million acts of science count. And so once you do the project, report it back to us on Citizen Science Month or .org or 1millionactsofscience.org, so we can count that as a part of your participation and get up to 1 million acts of science. So we'd love to have that involved. We do have another Do NASA Science Live event for anyone who is of interest of NASA Citizen Science, especially if you belong to the community of service members, veterans, and their families. We have an event coming up on April 10th that'll also be on the blog and the event database. So just a FYI for that. I mentioned this before. If you have any need for resources, we've got plenty on SciStarter. We can connect you to the right people too. So just reach out anytime. Our info@scistarter.org email is our best one. A bunch of us get those emails, including the 3 SciStarter folks on this call right now. Roland and Caroline and myself, so that we can get the answer to you when we can. But if you loved this event, feel free to let us know and respond to this survey. It takes like 2 minutes to do, just to say, great job. What else? Or I want to hear about something else. Whatever you want to know. We'll figure out how to get a topic on board and bring them into the fold. But that's about it. And we have 1 minute left if anyone wanted to have some last words to our audience to live in infamy in this recording. Or any questions or comments. Yes.
Thank you all for coming. And SciStarter, thanks for organizing these. It's really amazing to get to connect with people this way every time there's something cool to talk about.
I know. I love talking about something cool. So thank you. Thank you both for being with us and talking to us about these things.
Yeah, thank you. Thank you so much.
Awesome. All right. If there aren't any more questions, we will close the recording and say goodbye to Facebook and close the webinar. But we'll hopefully get a bunch more pictures in for you all about these weird locations with slime and sludge and grossness. I will be looking all over my apartment. It'll be very rude awakening. I'll try to prevent myself from cleaning it up right after. So if there's a chance it needs to be collected as a sample, it'll still be there.
That's perfect.
Awesome.